Now we've talked about how we read genes, what chromosomes are, and so forth. Next thing to look at is mutations. We've talked about mutations when we talked about evolution, and we know that that means that there's been an alteration to the genetic code. So there's an alteration to the directions on how to make an organism. And what I've got here in this picture are fruits and vegetables. And so I want you to take a look and see if you can identify any of them. So just take a little bit of time and see if you can identify any of these fruits and vegetables in this picture. All right, so let's take a look at them and see if you got any of them correct. So, there's one here that is not a fruit or a vegetable that's actually a fungus, and you might have caught that. So going across here, this one right here, that is an eggplant. So it's split into a bunch of lobes. This one, that is a pepper. We've got zucchini, cherry, which you may have actually come across one that looks like that. This is yellow squash, a carrot. This is a potato, a mushroom, so there's our fungus. Here we've got an apple. I know it looks bizarre, but it is an apple. If you look down here at the bottom, it's got the bumps on it. Then we've got tomato, cucumber, and then this one is either a nectarine or a peach. I'm leaning towards nectarine because it looks kind of shiny instead of fuzzy, but you can see that all of these have problems. Either they have doubled, like the cucumber and squash and zucchini, or they've got what look like growths, right? Like the cucumber, the tomato, the cherry, or they're missing parts like the apple. And these are all due to mutations. Now, are these mutations a problem insofar as these aren't safe to eat? No, right? These are all going to be safe to eat. We're generally not going to see these show up at a grocery store, but you might find something like this at a farmer's market. So mutations, like we've seen in a previous chapter, it's just the alteration of the sequences of the DNA bases. So we're changing the order of those A's, T's, C's, and G's in some way. And it can lead to changes in the structure and function of proteins produced. So we've seen now previously in lectures for this chapter that that's what our DNA is for. We're taking the information from those genes and we're reading it through making copies of mRNA and then taking that to the ribosomes to make proteins. Well, if we make any changes in those directions in how they're written, it's going to end up making changes in how those proteins are going to be built. And so since it's going to make changes to those amino acids, it's going to make a change to the shape and therefore how that protein is going to function. And this can have a range of effects. It can be anywhere from no change whatsoever, because maybe the amino acid is going to be the same amino acid, or it can make huge changes. It also depends on the alteration. Maybe it's only a single amino acid that's been changed, or maybe it's several, right? And so here is an example. Here's our friend the fruit fly again. And we've actually been doing a lot of work for a long time on mutating fruit flies. And here we've got our normal fruit fly face, eyeballs, antenna, the mouth parts. But this fruit fly on the right-hand side has been mutated. So they've changed the DNA so that instead of these normal antennae right here, the fruit fly grows eyes on that section on the end of the antenna instead of their normal antenna. So they've got their big normal eyes, and then they've got these little tiny eyes on the end of their antennae that would normally be, what they use to smell, right? And now instead they have eyes. So we've been able to do a lot of these sorts of mutations because we know the genome for the fruit fly really well. So we can go in and alter it, move information, for example, how to make an eye, into the area where the gene would be read to make an antenna, and boom, now we have an eyeball instead of an antenna. Now, these kinds of mutations show up everywhere. We have them in humans. So in a previous lecture, I had a question that had information in it about a gene for breast cancer. There are actually at least 2 human genes that have been identified that have a role in increasing breast cancer in humans, and they've been named BRCA1 and BRCA2. And these 2 genes are interesting because we have these genes, and when they started looking at these genes in women and men who had breast cancer, what they found is that it wasn't a single mutation that led to an increase in breast cancer. It actually was more than 200 different changes in those DNA sequences that have been detected, each of which results in an increased risk of developing breast cancer. So it's not just one alteration like we did with the fruit fly to make them grow eyeballs. It is 200 different changes in those sequences that can lead to an increased risk of developing breast cancer in these 2 genes. So this is why we can only talk about an increased risk, for example, on different types of cancers that we know that have genetic basis, because you may have, that increase because of these, but they may not ever actually be enacted. And so mutations, they have gotten a very bad reputation. Why have mutations gotten a bad reputation? So yeah, mutations have gotten a bad reputation because we usually associate them with things like cancer, right? Things that are pretty bad. Mutations tend to be disruptive, like cancer. They tend to create structures or problems that are problematic for the organism. And so they're changing things for the organism and causing issues. The thing is, though, believe it or not, mutations are actually very, very rare. You might be thinking, yeah, but I hear about, things like cancer all the time, and we do hear about it. But that mainly has to do with the fact that we're living long enough that these mutations build up. We actually don't get tons of these mutations that create problems. And we carry a lot of these mutations around, but they happen in problems that are in areas that don't cause problems. The mutation has to, if you remember back from chapter 10, it has to happen in a gene. So if it happens in that other 98% of your DNA that's not a gene, it's not going to be a problem. And the other thing is, remember, mutations are incredibly important. We need them in order to fuel evolution, right? So they are a requirement in order for evolution to occur. So there are a couple different types of mutations, and I just want to show you what these mutations might look like and what they do. So we're going to look first at what we call a group of point mutations. These are where there's changes to just the DNA. And then we're going to look at what we call chromosome mutations or chromosomal aberrations, which is where you actually have changes in entire sections of chromosomes, which also can lead to mutations. So what we've got here is a strand of our normal DNA, and you can see the bases up here, right? We've got GAC, ACA, and they're set into the little groups to code for amino acids. And then we've got the coding into the mRNA, so you can see the U's. The U's are pink in this one. And then what the normal protein looks like. And so we've got abbreviations for the amino acids, and they're color-coded so we can easily see what's going on here. All right, so let's take a look at the first type of point mutation. So what we've got first is what we call a substitution. This is just where you swap out one base for the other. So it looks like we're swapping what is that? An A for a T. So it was an A, now it's going to be a T. And that's one small change in the directions. So putting in a T in place, you can see up here we originally had the CYS, the one that got coded this green color for the amino acid. And now we've got this SER, the same one that's next to it. So this is going to change the function of the protein more than likely, because that one little change is going to change the amino acid and can probably change the shape of the protein. Now the other types of point mutations are a little bit more extreme, and that is because the first one is an insertion. So instead of swapping, what we're doing is actually putting in a new base. So in this case, we're inserting a C right here in front of this section of the original DNA. And what happens is all of the information just gets shifted down by one. What that does is changes even more. So with a substitution, we're just, in this case, changing one amino acid. But if you take a look, an insertion is going to change all of the amino acids from that insertion on, right? Because what it does is it shifts the letters along and changes the sets of the letters that are coding for these amino acids. And the same thing is true for a deletion. So you can have a deletion where one of the bases is removed and it shifts the other direction. So everybody rolls back one, and you get the same thing. You get new sets of letters to code for the amino acids, and so you'll get a completely different protein. So the substitution might make small changes to the protein structure and function, but insertions and deletions make major changes because it changes what we call the reading frame. Basically, it changes the letter sets for coding for those amino acids and changes all of these amino acids and will completely make a different protein from that point on. All right, so the other set I told you were chromosomal mutations or chromosomal aberrations. And this is where you get changes in the entire gene. So point mutations, you're going to be just changing small sections of the gene. But with chromosomal aberrations, we're going to change other things about the entire gene. So you can see here we've got 3 genes on a chromosome. And so the first thing you can do is just outright delete the gene, right? Take the directions out. That would be like if we're looking at our genome like a cookbook. This would be like somebody coming along and ripping the page out of the cookbook so you don't have the recipe anymore. Can't find the directions, can't make that protein, right? The other option is gene relocation. This would be like somebody coming to your cookbook, ripping out the page and sticking it in another spot in the cookbook. You don't know now where to find the directions, and your cells have that same problem. So gene relocation, you can see that the gene 1 has been picked up and moved to another chromosome. And remember, we talked in a previous video that the chromosomes are like chapters in the book, like chapters in a cookbook. And so this would be like if you have a cookie recipe that would normally be with all the other cookies, but you take out that one cookie recipe and you stick it in with the appetizers, right? With like the dips and things like that. You wouldn't be looking for a cookie recipe with your appetizers. And so this gene will likely never be read to make that protein or proteins that it makes because it can't be found. The last option is duplication. So an entire new copy of the gene can be made. So now there's more than one copy. Sometimes this is good. This has actually occurred to do things like allow us to dissolve amylase or starch. We have multiple copies compared to other primates. That's why we can eat more starchy foods. However, very often this is a problem because more than one copy is going to make more of whatever that gene does. And so you're going to be reading that gene, and so you're going to end up making more of the protein or, whatever product it is. So very often, almost all of these are proteins, but say maybe it's an enzyme. One of those proteins that takes stuff apart and puts stuff together. You have too many of those guys around, they might start taking things apart that they're not supposed to. All right. So here, just to give you an idea of what these mutations might look like as far as if it was written into sentences. So we've got the original sentence, one big fly had one red eye, talking about fruit flies. So if we have a substitution, this is where you slop one of those letters out. This is what it would look like. Most of it's still okay, like I was pointing out, but you've just got that one little change. So you can mostly still figure out what it's talking about, but a little bit of a problem. Here, this is where you would actually end up deleting sections of it. I didn't show you that one, but this is a mutation that can happen as well. This is a frame shift. This is where you get that insertion or deletion that shifts all of the bases one way or another and changes all the amino acids. And so you can see here's where that insertion or deletion happens. And you can read just fine up until that point, and then it just makes no sense afterwards. And so that's why those kinds of mutations make such a big difference. And then you can see some of the other kinds that you can get. So you can delete multiple sets of letters. So this is a deletion of 3 sets of letters or one whole amino acid set. And so you have one big, and now you don't know what, had one red eye. And so there are other guys here where you can duplicate a section. So that would be almost like duplicating a gene, inserting a new section. And then this is a whole nother one. I'm not going to go into that. If we were doing more genetic information, I talk about that expanding repeat. All right, so I end up in this section with question, mutations have which of these? A negative effect on the gene product, aka protein, a neutral effect on that product, or can cause beneficial changes in the gene product or its expression. Or is it a combination of any of these? And in this case, it is all of the above, right? Mutations can have any of these effects. We think about beneficial ones when we think about like evolution and natural selection. We think about negative effects when we think about things like cancer and broken enzymes. But it can also have a neutral effect because you can get a mutation that doesn't make any actual change to what you're making. So some of these amino acids actually are coded for several different sets of letters. And so you can swap out the sets and it just is going to make the same amino acid set in that protein.